Hello and welcome to today's USC Viterbi Live featuring Dr. Stacey Finley. My name is Tiffany Tay and I am the Executive Director of Alumni Relations and Annual Giving here at the USC Viterbi School of Engineering. We started this Viterbi Live series about 3 months ago to bring relevant content to you, our engineering community, and I hope you're able to join us for a few. If not, we have them all recorded and posted online on our online event series page, which we'll paste in our chat box momentarily. Throughout the presentation, please use the Q&A box to submit your questions, and Dr. Finley will answer them all at the end, time permitting. Today, we are actually taking a break from our regularly scheduled COVID-19 content to highlight one of our superstars in our engineering department and the research she's doing in a field that continues to be persistent, cancer. She will share how she uses mathematical models to decipher the complex networks of reactions inside of cancer cells. Dr. Finley is an associate professor in the Department of Biomedical Engineering here at USC. She is the holder of the Gordon S. Marshall Early Career Chair and the director of the Center for Computational Modeling of Cancer. Dr. Finley also has a joint appointment in the Mork Family Department of Chemical Engineering and Materials Science and the Department of Biological Sciences. She is also a member of the Norris Comprehensive Cancer Center. So please join me in welcoming to the Zoom stage, Dr. Stacey Finley.
Okay, let me get the slides up. All right, thank you very much, Tiffany, for the introduction and really for this opportunity to be able to talk a little bit about my research and the work that I do here at USC in the Department of Biomedical Engineering. I'm really excited to tell you about how we're using mathematical modeling to better understand cancer cells and to help improve cancer treatment opportunities. So this work is really situated in the broader field called mathematical oncology. This is the use of mathematics to understand cancer and help develop better treatments. I am a quantitative person. I'm really fascinated by numbers, and I get to indulge my love for numbers in an impactful way through my research. Using math, I can probe the complexities of cancer, and in particular, I study complex networks inside of tumors. These networks influence how the tumor responds to treatment. Now, I'm willing to bet that each of you also tries to optimally understand a complex network. For example, you've probably navigated a network just like this many times, maybe not so much recently with the quarantining, but still, I'm sure it's fresh on your mind. You want to be able to understand how much traffic there is on each road and how long it would take you to get to your destination. Now, similarly, there are complex networks inside of cancer cells that we must optimally navigate. This is an example of a network that allows cells to grow and to thrive. And this allows cells to produce energy. So today I want to tell you about some of the work in my own lab to study networks that allows tumors to gain their own blood supply, and also networks inside of immune cells that can be harnessed for immunotherapy. So the overall goal of our research in my lab at USC is to be able to predict the dynamics of biochemical networks, these complex networks in cancer, and use that understanding to help develop new drug treatments and also better understand the effects of existing drugs. And the tools that we use to accomplish this goal are called systems biology approaches. And this is where we study the whole, not just individual parts. And we do this using mathematical modeling combined with experimental studies. So some of the networks that we're really excited to study first have to do with immune cells and how the immune cells will become activated and how that activation can be harnessed to target cancer cells. Another set of complex networks that we study are the networks that allow the tumor to gain its own blood supply. And we've published some papers in this area as well. And then we also look at cancer metabolism. So that's the example of the network that I showed a few slides ago, which allow a cancer cell to get the nutrients that it needs and to metabolize those nutrients to produce energy needed to fuel its own growth. So given the limited time that I have today, I'm going to tell you two stories. The first has to do with immune cell signaling, and then the second has to do with complex networks related to the growth of new blood vessels. So in the context of immune cell signaling, we're studying a type of receptor, an engineered receptor called a CAR, or chimeric antigen receptor. This is an engineered receptor that can be expressed on the surface of T cells, and it allows that T cell to be able to target and bind to, whoops, I think I might have to go back 1 second. But it allows a T cell to be able to target and bind to a tumor cell. And in doing that, then the T cell becomes activated. Let me go ahead and share my screen again. All right. I apologize for this. Let's see. Okay, so we're talking about CAR receptors. They can be expressed on the surface of T cells. These are indigenous cells that are already residing in the body that can be harnessed to target and kill a tumor cell. And that CAR allows that to happen is because it's comprised of an extracellular, so that's outside of the cell, that would bind to an antigen, a tumor-associated antigen, that's TAA here at the bottom, that's presented on the surface of the tumor cell. And it's combined with a number of intracellular signaling domains that are derived from the endogenous T cell receptor. And CAR engineered T cells have been very effective in different types of cancers, even leading to the complete remission in B-cell lymphoma. And this is really exciting. It's really pushed the field forward in terms of using a patient's own cells, engineering those cells to express the CAR, giving those cells back to the patient, and allowing the cells to target and kill the tumor cells. There still can be some serious effects that happen when T cell activation goes awry. So I want to just list a couple of those limitations. The first is called cytokine storm. This is an overactivation of the immune system that is perhaps thought to occur because the CARs do not have all of the control mechanisms that endogenous T cell receptors have. And thus the immune system can be overactivated, flooding the body with these cytokines, really have very serious side effects for the patient, even leading to fatalities. Another limitation is what happens when we try to adapt the CAR therapy to other types of cancers. So the cancer that I mentioned where this type of treatment has been very effective is called B-cell lymphoma and other cases. But in this particular case, the antigen that the CAR is trying to target is called CD19, and it is only expressed on the disease cells. And that same level of specificity is not present in other types of cancers. For example, HER2 is highly expressed on certain breast cancer cells, but it's also expressed on healthy tissues. And so then the CAR is doing its job in targeting that antigen, but it's having off-tumor effects. And these can also lead to serious side effects for the patient. And so what has is to try to better control that signal and control the activation of the T cells by adding additional intracellular signaling domains. You can see that with the first and second and even third generation CARs shown there, as well as being able now to target multiple molecules on a tumor cell to increase the specificity so that the CAR is really targeting just the tumor cells, the disease cells, and then also combining those together, so multiple targets and better control with chimeric inhibitory receptors. And so this has been effective, but it's really pursued in sort of a guess and check or trial and error sort of way. There's no fundamental and systematic study of how the structure of the CAR may influence T cell activation. And so that's really where we believe mathematical modeling can be used to better understand T cell activation and aid in the design of CAR T cells. So I'm going to walk you through the complex signaling network that happens when the CAR recognizes and binds to an antigen. In this case, we'll look at the CD19 antigen. So what I'm showing here is a schematic of a second generation CAR. It has the CD28 costimulatory domain that's right there at the top. It has the CD3 zeta, which is closer to the bottom. That CD3 zeta is actually comprised of 3 different domains, 3 ITAMs, A, B, and C. And the circles there are just sites where that molecule can be phosphorylated. So I'm sort of taking you back to biochemistry, where hopefully you remember that when a phosphate group is added to a molecule, that can lead to a whole cascade of signaling events and allows the molecule to become activated. So when there's a P there inside of that circle, that means that that site is phosphorylated. Now, another key player here in this signaling network is called LCK. This is an enzyme that catalyzes the phosphorylation of these different sites. LCK can actually undergo autophosphorylation, so it phosphorylates itself, and then it mediates the phosphorylation of the different sites on the CAR structure. Now, once 2 of the sites on an ITAM are phosphorylated, then another molecule comes in and binds. This is called ZAP70. LCK can also phosphorylate ZAP70. Now, these are all activating events that happen once the antigen is bound. There are also molecules that are opposing these activating events. One is called a phosphatase that's called SHP1. It's actively aiming to remove those phosphate groups and shut down the signaling. But given strong enough binding of the antigen, long enough binding of the antigen, then the cell can overcome that negative feedback and lead to the formation of this multi-protein complex called the LAT signalosome. And this ends up leading to signaling through the MAPK pathway. This is a pathway that's found in many different types of cells. It leads to activation of ERK, so that's there shown on the bottom in this green circle. That is a protein that, or a transcription factor that allows the cell to promote its own proliferation. And ERK or ERK can also provide some positive feedback in order to prevent the action of that inhibitor called SHP1. And then we also have 2 other molecules here that are shown right there binding to CD28 as ways in which CD28 can augment the signal, this co-stimulatory domain. Now, I actually want to emphasize that this is a simplified schematic of the pathway. It includes a lot of different biochemical reactions, positive feedback loops, negative feedback loops. And so it's not immediately clear how changing the structure of the CAR would influence the level of activation of the T cell. And so that's where we believe mathematical modeling can really help us to better understand this complex signaling network. So I'm just briefly going to talk about some of the methods that we use. I'm happy to go into more detail. I know the audience is coming from a pretty broad background, so I'm definitely excited to talk about the details of our methodology. But for now, let me say that we use a rule-based modeling approach that allows us to automatically generate all of the reactions that comprise that schematic that I showed on the previous slide. It gives us a set of ordinary differential equations, so now taking you back to calculus, and those differential equations will describe how the species concentrations evolve over time. Now, with any mathematical model, the values of the parameters are very important, and so we need to make sure that the parameter values are set properly so that the mathematical model can match the experimental data that's available to us. And so we estimate the values of any parameters that we can't measure directly or that we can't take from the literature. We estimate the values of those parameters by fitting the model to some experimental data. And in the process of that fitting, we find the optimal parameter values. So the way that we do that, we use an algorithm called particle swarm optimization. But here is sort of a takeaway example of how we can fit the model. So the circles on all of these 4 plots are experimental data that we've extracted from the literature. And the lines are the fit for our mathematical model. So you can see that we do a pretty good job of matching. What's shown here is the percent phosphorylation of these different proteins as a function on the x-axis of the concentration of that some phosphatase called CD45, so another protein that aims to remove those phosphate groups. And so you can see that the lines are matching pretty closely to the experimental data points. And then on the right-hand side, you can see some of the parameter values that we've estimated. So we're not actually trying to estimate hundreds of parameter values. It's really a small handful of parameter values, and those are the optimal values that we get from this model fitting. Most of them have very tight confidence intervals, which means that the modeling fitting approach is very robust. It gives us confidence that we've identified the optimal parameter values. Now, with this optimized model, then we can use it to answer some specific questions related to T cell activation. So the first question that I'll walk you through is answering this question about what is the role of CD28? And then the second question is trying to understand what influences the population response. So for this first question, this is really work from my first PhD student, Jennifer Roars. We published it just recently, earlier this year, and also a couple of papers over the last couple of years. But what we're trying to understand here is how does CD28 affect T cell activation? And in particular, we're characterizing T cell activation by looking at the ERK response time. So it's been shown in the literature that the time that it takes for 50% of ERK inside of cells to become phosphorylated is a measure of the activation of the T cell. And so what we do is to use our mathematical model to predict the ERK response time, and that's what I'm showing on the vertical axis, as a function of the antigen concentration. So increasing antigen concentration means greater stimulation of these engineered CAR T cells. So what the model predicts is the level or the time that it takes for 50% of ERK to become phosphorylated at different antigen concentrations. The blue line is what happens when we have a CAR that only consists of that CD3 zeta chain. And then the red dashed line is what happens when we have the CD3 zeta chain combined with CD28. So we're again trying to understand what is the role of CD28 and how does it influence T cell activation. It might seem like a very fundamental question, and indeed it is, but this type of fundamental understanding of the roles of the different co-stimulatory domain really hasn't been addressed in a lot of detail in the literature so far. Again, I mentioned earlier that the design of CARs is really based on a guess and check or trial and error approach. And so by having more fundamental mechanistic understanding, we can help in the design of more optimal CARs. So the model predictions are showing that the presence of CD28, so the red line, is below the blue line for all the different levels of stimulation that we simulated. So it means that adding CD28 is speeding up the response time. So it's shortening the time that it takes for the cell to become activated. Now, this is a novel model prediction. We wanted to test and validate that model prediction. So we turned to an experimental system, and Jennifer also did these experiments where she engineered T cells to express the CAR. She stimulated those CAR expressing T cells with a cell that's expressing the CD19 antigen so that we could really simulate this cell-cell interaction. And then she measured with flow cytometry the ERK response time. And so these are the experimental measurements that we got from Jen's work, where again, I'm showing on the vertical axis the ERK response time. On the horizontal axis is a measure of the level of stimulation. And we were really excited to see that the experiments do indeed validate the model prediction that having the CD28 co-stimulatory domain speeds up the response time, so it lowers the ERK response in minutes. And it allows us to better understand this role of CD28. Now, I can go into a lot more detail to try to understand the way in which CD28 does this. And we have some hypotheses about that. We've also, we're working on testing those hypotheses about what is the mechanism by which CD28 enhances the ERK response time. But what we get from this mathematical model is that we can better understand the effect of CD28 on T cell activation. So that leads me to the next question in this area of studying immune cell signaling, and that is, can we better understand the response of a population of cells? Now, this is relevant because in T cell therapy with CAR engineered T cells, this is called adoptive cell therapy, where a patient's own T cells are extracted from the body. They're engineered outside of the patient's body to express the CARs. And then that population of cells is presented back to the patient so that the cells can target the tumor cells. So it's very important to understand the response of the population, because that is indeed what the patient is receiving, a population of cells. That cell, each cell is not going to be the exact same. It's not going to be a copy of one another. There's going to be variability in that cell population. And so we wanted to understand, how does that variability influence the response to being stimulated by the antigen? And this is work by Colin Chess, who is a current PhD student in my lab. So what we did is to simulate a population of 100,000 cells. Now we're running our model 100,000 times. Each simulated cell has a different amount of the starting material, the starting proteins inside of the cell. Each cell also has a different level of expression of the CAR. We know that there's going to be this variability in the population of T cells, and so we try to implement that same level of variability using the mathematical model. So what I'm showing here is an example of the results that we get from this simulation of a population of T cells, CAR-engineered T cells, where now I'm plotting on the vertical axis the percent of phosphorylation of ERK inside of those cells. And now on the horizontal axis, it's over time. So this is the dynamics of the ERK phosphorylation. Now I'm just drawing a line right at 50%, because that's how we're going to categorize a low ERK response compared to a high ERK response. But first, what you can see in this representative sample of 100 cells is that there's a lot of variability, right? So right over towards the left-hand side of the figure, you go from 0 phosphorylation all the way up to 100% phosphorylation very rapidly. There are some traces, some of these lines that don't get all the way up to 100% phosphorylation, some that maybe get to just less than 10%. And so having that variability that we again see in clinical samples shows that there is now a difference in the amount of ERK that becomes phosphorylated, even though the cells are all being exposed to the same level of stimulation. So what we wanted to do is to try to understand, can we identify which cells would lead to a high ERK response and which would lead to a low ERK response solely based on their initial conditions, because that's something that's measurable. That's something that we can quantify before presenting the cells back to the patient. And so if we can classify this cell response just based on the initial concentrations, it would be a way to enhance the patient's response to CAR T cell therapy. So what we did to answer that question is use a data-driven modeling. It's sort of machine learning light, called partial least squares regression. So it's relating the inputs to some outputs. In this case, the inputs are the things that we're changing and representing heterogeneous population of cells. So we're changing the amounts of the initial concentrations. The output is this classification as to whether the ERK response is high or low. So we're trying to understand how those inputs are influencing the ERK response time. So we can do PLSR. We get out a measure of a way to classify the cells. And you can see that just looking across the horizontal axis from left to right, we go from cells that have a high ERK response, and that's shown in blue, to cells that have a low ERK response, and just moving from left to right. So then the question is, what is changing for each cell as you go from left to right? Can we identify which initial concentrations are most influencing this classification of the cell of having a high ERK response versus a low ERK response? So we can dig more into our PLSR, our partial least squares regression analysis, to identify the key or influential inputs. So one way to do that, one measure of the influence, is called the VIP score, variable importance of projection score. So what I'm showing here is the VIP score for each of the species whose concentrations varied over time. And we can see that there are just 6 bars that are highlighted in orange. Those are for the initial concentrations of species that have a VIP score greater than 1 based on our PLSR analysis. And a good rule of thumb is that a VIP score greater than 1 means that that input is significantly affecting the output. So there are 6 of these initial concentrations that are directly influencing whether the cell will have a high ERK response level or a low ERK response level. And just to bring your attention to this bar all the way to the right, it's ERK. So it's interesting that the initial amount of ERK that the cells start with is not a key influencer as to whether the cell will have a high or low ERK response. So this is not immediately intuitive. We really do need this kind of data-driven modeling approach in order to identify what are the key inputs. Now for each of these 6 key inputs, these are the dark bars that are shown on the right figure, we can understand in which direction would that input pull or push the response level. So for the 6 bars, those are highlighted with the darker color. If it's red, it means that it would push it towards having a low ERK response level. If it's blue, it means it would push it towards having a high ERK response level. So one thing that was really of interest to us from this analysis is that the amount of CD3 zeta, again, that's a component of the CAR itself, would actually push the cell population to have a lower ERK response. So increasing the amount of expression of the CAR would actually decrease the ERK response level. Again, this is counterintuitive. We wouldn't immediately guess that that is the case, but with our very detailed mechanistic model combined with this data-driven model, we can try to understand why the prediction is this way. And so what we did is to vary the amount of CD3 zeta and look at the ERK response. This is the amount of phosphorylation as a function of time. As you go from the very bright green to the dark blue, so from left to right, it's increasing the CD3 zeta. And so what you see is that having greater expression of the CAR would actually slow down the time that it takes for the ERK to become fully phosphorylated. And at the same time, we can also look at what happens to the phosphatase. So just a reminder that the phosphatase is that molecule that tries to oppose the signal. It tries to remove that phosphate group and inactivate the CAR. And so going from the bottom to the top, so bright green to dark blue, is increasing CD3 zeta. And so we see that as you're increasing and having greater expression of the CAR, it actually leads to higher levels of this inhibitory molecule, the phosphatase level. And so that's the way in which we see that it's not important, it's not just important to increase the amount of the CAR that the cells are expressing. We need to be able to balance that with the opposing negative feedback that's happening inside of the cell with this complex signaling network. So just to summarize this first part of the talk in studying immune cell signaling, what I've described so far is that we constructed a mechanistic model of CAR T cell activation. We can use that model to distinguish the effects of different CAR constructs. We can use the model to simulate a very heterogeneous population of CAR engineered T cells, and then be able to identify which aspects of the cell, the intracellular signaling species, which aspects would influence the activation of the CARs themselves, the CAR T cells. So we're combining mechanistic mathematical modeling with data-driven mathematical modeling in order to gain better insight into how to modulate the cell response to enhance CAR T cell activation. This is exciting to us. We're actually moving forward with studying even more about T cells, now not just looking at ERK, which is influencing proliferation, but also looking at other signaling molecules inside of T cells, such as the molecules or proteins that would influence cell survival. So this is another important aspect of CAR T cell activation and the efficacy of these therapies, that we need the cells to persist in the body to survive. And so it's also important to look at signaling that influences cell survival. And we're doing that right now. So I'll switch gears and tell you a little bit shorter story about a complex network that influences how a tumor will gain its own blood supply. This is called angiogenesis. So angiogenesis really does drive tumor growth. The tumor needs its own blood supply in order to continue proliferating and to grow and to thrive, just like any normal tissue inside of our body would need a steady blood supply to receive oxygen and nutrients for growth. The balance of promoters and inhibitors of this process, so proteins that would promote new blood vessel formation compared to proteins that would block or inhibit new blood vessel formation, there's a balance in these cases when there's no new blood vessels forming. But as the tumor continues to grow, then there's an overabundance of the promoters. And so this means that there actually have been a lot of cancer treatment strategies to block the formation of new blood vessels by targeting these promoters. In my lab, we're actually looking at the other side of the balance. We're looking at the inhibitors, and can we use mathematical modeling to predict the effects of different strategies to mimic the inhibitors of new blood vessel formation. And one particular protein that we've been focused on is called thrombospondin 1, or TSP1. And I'm just going to walk you through another complex network that we get very excited about to try to better understand the effects of this inhibitor of angiogenesis. So I'm showing here a promoter. It actually has a specific name called VEGF, but it promotes cell growth. It promotes cell migration by binding to a receptor that's on the surface of endothelial cells. And this inhibitor, thrombospondin 1, aims to block the signaling that this promoter will mediate. And this thrombospondin 1, this inhibitor, can also promote cell death. So these are multiple mechanisms by which thrombospondin 1 can inhibit the formation of new blood vessels. TSP1 can also trap the promoter, so it can bind directly to the promoter. It can allow the promoter to form these non-signaling complexes as another means for inhibiting the formation of new blood vessels. And then also it can directly block the promoter and reduce the availability of that promoter for forming new blood vessels. So again, we get excited when we see this kind of network because I want to know, what are the dynamics? How over time do things change? If we want to come up with the best strategy for inhibiting the formation of new blood vessels, which aspect of thrombospondin 1's mechanisms should we take advantage of? And those are the types of questions that we can answer using a mathematical model of this complex signaling network. So we have created a mechanistic model of thrombospondin 1 in tumors, and now we're using it to understand at the whole body level the balance of the promoters and inhibitors. So just to provide a little bit more context, targeting just the promoter or mimicking just the inhibitor, this single agent therapy strategy has had limited success. It is possible to combine a strategy that will mimic the action of the inhibitor and block the action of the promoter. But even with this dual inhibition treatment strategy, it did not show a lot of efficacy in all patients. So there was actually a lot of variability in the response to this dual inhibition treatment strategy. And so that actually got us excited because we thought, well, maybe we can use mathematical modeling to better understand why this heterogeneity in the response exists. And so we applied the model to try to see, is there a set of biomarkers that can predict the treatment efficacy? So this is the specific biological question that we're using the model to answer. So the model that we're actually, that we have already created is representing the entire body of a cancer patient. It includes the tumor compartment, it includes all the normal tissue in the body, and then it also has the blood compartment to allow transport between different tissue compartments. And so we're using this model to look at the effects of this dual inhibition treatment strategy, where we have one drug that blocks the promoter. It's given once every 2 weeks. We have another drug that mimics the inhibitor, and we're using the same doses, the same timing intervals for the doses as what was used in that clinical trial, where only about 30% of patients actually responded to the treatment strategy. So we're really using the model to simulate actually a cohort, a virtual cohort of 1,000 patients receiving this dual inhibition treatment strategy. And the way that we have 1,000 unique patients is by varying the properties of the patient's tumor. And so specifically, we're varying the levels of receptors that are in the tumor. And this is one aspect of heterogeneity that certainly exists from patient to patient. We could think about other aspects of variability across cancer patients, but this is one characteristic of cancer patients in having a different receptor profile. And so we simulate 1,000 unique patients by varying their receptors in the tumor cells. And then we predict the amount of VEGF, which is the promoter. We predict the amount of thrombospondin 1, which is the inhibitor. And we also look at the ratio of the two. So this is some of the results. I'm just going to walk you through a little bit of the results we get from the mathematical model. What I'm showing you now is what's happening inside of the tumor. The first figure at the top is how much VEGF there is as a function of time. The second figure is how much thrombospondin 1 there is, that's the inhibitor, as a function of time. And then the bottom figure shows the ratio of the two. Now, we first allow the model to come to steady state, and then we begin treatment, the simulated treatment. And that's indicated by the vertical dashed lines. And we simulate a 2-week cycle of treatment. And so that's indicated by the dashed vertical line on the right-hand side. The solid line within each box actually shows the mean value of our 1,000 virtual patients. The shading in each box shows the standard deviation. So you can already see that because we've varied the amount of the receptors that each tumor cell has, we are seeing some variability in the amount of the inhibitor, the amount of the promoter, and the ratio of the two. This is just what's happening in the tumor. We have similar figures for what happens in the normal and the blood. But what we wanted to do, instead of looking at all of these figures and what happens over time, we wanted a more concise way to quantify the response to treatment. So what we did is to focus in on the tumor and say, well, what is the fold change? So how much do these quantities change as a result of the treatment? So we have this fold change, which is the post-treatment level compared to the pre-treatment level. We have this ratio that we can then say, how much does the VEGF concentration change? That's the promoter. How much does the inhibitor concentration change? That's thrombospondin 1. And how much does the ratio change? So then we can say, well, if that ratio, the fold change, increases, then it would be in the dark gray area here. If it decreases, it would be in the light gray area. Nothing really from our mathematical model, nothing really exciting is happening to the inhibitor. In the tumor, in the blood, and in the normal tissue, the amount of the inhibitor is staying pretty constant, so right at 1. But the promoter and also the ratio of the promoter to the inhibitor, those are quantities that are decreasing due to the treatment that we've simulated. And what's interesting is that specifically in the tumor, that's the red box, we see a high level of variability. So this is again showing the variability for the response across the 1,000 patients that we simulated. There are even some outliers. Those are the circles, the red circles shown at the bottom. The variability is also shown by those long vertical red lines above and below the red box. And so we see that there's a wide range of responses due to the variability of each patient's tumor. So we wanted to go in, again, using PLSR, using this data-driven approach. Can we identify what are the influencers of that response to treatment? So again, we're looking at the VIP score. Again, a VIP score greater than 1 means that the amount of that input, in this case, the amount of the receptor, would be significantly influencing the response to treatment. Just first to show that on the tumor endothelial cells, so these are endothelial cells inside of the tumor, the receptors on those cells really, it doesn't influence the response to treatment. It's really the receptors on the disease cells, the tumor cells themselves, that are influencing treatment. And specifically, we found 3 sort of markers or predictors of response. The first is called VEGF. So this is a receptor that the promoter binds to. And so we can see how does the amount of that predictor influence the effect of treatment. So I'm showing here on the vertical axis, the effect of treatment. On the horizontal axis, the amount of the predictor. The predictor 1 is VEGF-R1. The predictor 2 is a receptor that thrombospondin 1 binds to. The blue dots in each of those figures are for each individual 1,000 patients that we simulated. The red line in each of the figures is just to sort of draw your eye to the shape or the trend of the curves. And so what we find is that when VEGFR1 is low, when CD47 is low, then the patient will have a better response. And for the third predictor, it's CD36, another receptor that thrombospondin 1 binds to. In this case, when CD36 is high, then the patient would have a better response. So we're really excited because we think that this might be a panel of 3 biomarkers that together would predict whether the patient would have a good response to this treatment. And this is really important because it would be helpful to identify those patients before they undergo treatment, before they are exposed to the anti-angiogenic treatment strategy, to identify which patients would actually have a positive response. So we're excited about this, and it helps us to understand that the heterogeneity in the response is due to variable receptor expression, and we can go on and identify which of those receptors that are varying will influence the response to treatment. So just to briefly summarize this part of the talk, I've told you about a whole body model that we constructed that allows us to predict the concentrations of different angiogenic factors inside of the body. We can use the model to predict the response to anti-angiogenic treatment strategies, and then identify using data-driven modeling which tumor-specific properties would influence the response to treatment. So we're moving forward with this work by trying to validate it using published clinical studies that we can really do a retrospective analysis of and see, does it validate what the model is predicting? So with that, I'll just wrap up to tell you that I've talked to you about immunotherapy work that we're doing in our lab, the study of blood vessel growth, and also what I didn't have a chance to tell you about is metabolism. But we're also collaborating with a lot of different researchers, both computational researchers here at USC, as well as experimental researchers in the Keck School of Medicine and in Dornsife as well, to really, again, validate and test our model predictions and to have this more holistic and integrative approach to better understanding cancer. I would be remiss if I didn't acknowledge the phenomenal students in my lab and trainees in my lab who have made this work possible, along with our collaborators and our funding sources. Today, I told you about the work of Colin and Ding, as well as Jennifer Roars, and our collaborators on the 2 projects are Pin Wong, as well as Nicholas Graham, both at USC. And we've had very generous support from federal funding agencies, as well as private organizations. And with that, I'd like to thank all of you for your attention, and I'd be happy to answer some questions.
Thank you so much, Dr. Finley. That was really remarkable information, and your excitement for all of the progress really shines through. So thank you again for that. So we did have a few questions come in. First one coming from Russell. How does the dose of CAR T cells affect the therapeutic efficacy, and is it cancer type specific?
Right. So I will say that the modeling approach that the model that we've constructed is sort of agnostic to the cancer type. We're focusing on the intracellular signaling network itself. Certainly that network, those network dynamics could change depending on the specific type of cancer. I think probably what's more relevant is which antigen would be presented by those cancer cells and how tightly the CAR would bind to the antigen. And so that's something that we can incorporate into the model really straightforwardly and try to see how that would influence the amount of stimulation. We haven't varied very much the dose in terms of the number of cells that would be returned to the patient, more so looking at the activation of the CAR structure itself along with the intracellular signaling domains. But these are certainly things that we're thinking about and that I feel it would need sort of a complementary modeling approach to the detailed signaling model that we have right now.
Got it. Thank you so much.
Ross.
Our next question comes from Lucas. How do you define post-treatment levels if the angiogenic ratio is oscillating?
Yeah, so the angiogenic ratio is oscillating mainly because the inhibitor of angiogenesis, so mimicking that inhibitor, that drug has a very short half-life. So this is ABT 510, it's produced by Abbott. It has a very short half-life, so it's actually being administered twice daily compared to the inhibitor of the promoter, which has a longer half-life, is only given once every 2 weeks. So that's why you saw those very short oscillations in terms of the time over which the oscillations happened. And it all has to do with the pharmacokinetic properties of the drug itself. But what we're actually saying is that over a single treatment cycle, which we're simulating for 2 weeks, that's the same treatment cycle that was used in the clinical trial. So we're just taking what happens at day 14 compared to what happened at day 0. And that's how we calculate the fold change. But you're certainly right that the angiogenic ratio is oscillating, and it has to do with the properties, the intrinsic properties of the drug.
Got it. Thank you so much.
Lucas.
All right, we have a comment and a question coming in from Heiko. The comment is, very fascinating work and excellent talk. The question is, for the third part of the talk on the angiogenesis modeling, I was very interested in the 3 predictors, and I'm wondering if you were able to evaluate these as a triple biomarker in an independent in silico or in vitro cohort.
Yeah, so first of all, thanks Heiko for listening in and for your very kind comment. For the question, yes, so that's what we're working on now. We're trying to, I think what we want to start doing is to go to the clinical literature where maybe it's not been quantified at the single cell level, like how many CD47 receptors there are, how many CD36 and VEGFR1 receptors, but more so looking qualitatively with immunohistochemistry samples that we can say, is there high expression versus low expression, maybe versus medium expression, and then use those clinical observations combined with, right, they would have to be paired with the treatment strategy itself. So this is something that I think is lacking in the literature. We've tried to find as much as we can, but you're right. I think we need to now maybe do our own experiments or collaborate with the group to do the experiments, first starting in vitro to see, to really validate, does the level of those receptors relate to their responsiveness to the anti-angiogenic treatment strategy. This is on our list. It's really exciting because we have this exciting, perhaps, you're right, panel of 3 biomarkers. And so we want to identify whether and validate whether that is reliable or not.
Thank you so much, Dr. Finley. All right, our last question is from a recent grad breaking into the industry. I just completed my PhD in physics bioinformatics. Do you have advice for someone who is strong on the mathematical side, but weak on the oncology side to break into this field?
That's a really great question because I feel like that was me. So my degree is in chemical engineering, and I grew up, like my training has to do with heat exchangers and distillation columns, like nothing to do with biological questions. And so it's really only in my postdoc training, my postdoc fellowship at Hopkins, where I was able to almost immerse myself into the biological side. That's where I learned all about angiogenesis and cancer. And it's really been since starting my postdoc that I've expanded my understanding of the biology and physiology as well. And so I personally think, and maybe there are people who would disagree, but they don't have a stage, so I can say this. But I personally think that starting with a quantitative background and moving into this field really positions you well, because you can ask the more hardcore math questions. You have a physics background as well, so you can make sure that the models that you would generate are physically sound and reasonable. And then I just suggest that you read as much literature as you can on the biological side and on the physiology side. I think that there are probably people on this call now, and I can point you to other resources of people who are looking for postdocs in this area that have this type of training. And I feel like you can pick up a little bit more easily the biology that you need, because you're just going to be trying to understand as much as possible the context in which your mathematical model is going to be grounded. And so you're going to want to learn as much as you can. and you'll have the foundation of the math on your side as well. So I would suggest just reaching out to researchers in mathematical oncology that are really actively looking for probably someone with your background.
That was wonderful. I think that brings us to the end of our Q&A and the end of our webinar. Thank you so much for being here today, Dr. Finley. It is always a pleasure watching you present, not only on this, but for the career networking advice too. So we truly appreciate you. And thank you to all of our participants for tuning in. This session was recorded and Dr. Finley has generously agreed to share her slide deck as well. So all that will be posted on our online event series page within the next 24 hours.
Great.
So thank you again for joining us today. And as we say here at USC, fight on everyone. Have a great rest of your day. Have a wonderful weekend. Thank you, Dr. Finley.
Thank you. Thank you all for attending.